Our first example and the first speaker is Austin McLaughlin. I will not reintroduce him because you just heard about his curriculum. I will just mention one thing and highlight this one, that he is as well very much involved in the Biosafety Clearinghouse, and this is the first database which will be presented today. Austin, please.
Thank you very much, and thank you again for allowing me to speak twice. I feel like that's a special honor for me. So today I'll be speaking about our Biosafety Clearinghouse that the Secretariat hosts. So the Biosafety Clearinghouse, or BCH, was established by Article 20 of the Cartagena Protocol and as part of the clearinghouse mechanism under the Article 18, Paragraph 3 of the Convention on Biological Diversity. The BCH is an instrument that allows parties to exchange information on living modified organisms, or as also known as GMOs, and are therefore a fundamental role in facilitating the implementation of the Cartagena Protocol. The BCH fosters transparency in the regulation of LMOs, what rules apply, whom to contact for more information, in decisions taken on LMOs, what LMOs have been approved or prohibited, for what uses and which jurisdictions, and access to information. And the BCH really is for everyone. It's not only for parties, but it's also for general users, as well as non-parties. Governments that are not parties to the Cartagena Protocol are also encouraged to publish information on the BCH. A large number of the decisions and information on the BCH do come from these parties. But other stakeholders can also publish information on the BCH, and some types of this information is made publicly available. The BCH itself is sort of split into two types of records. We have our national records, which are published by governments and include information that parties are obliged to provide in accordance with the protocol, as well as other national information that might be relevant for the implementation of the protocol. And then we also have our reference records. So these are biosafety-related resource records and scientific information. This information is submitted by users of the BCH and are validated by the Secretariat. So as a sort of a schematic, we have several inputs that go into the BCH. We have our national authorized users, the Secretariat. BCH users input information into the BCH, and all of this information is made freely and publicly available. So the BCH itself does contribute to the field of detection and identification. It's a database that contains a lot of information related to biosafety, so scientific records, biosafety experts. We have a new platform which has an improved user interface, so we have a better search mechanism within the BCH, as well as cross-referencing between records. The BCH links to other databases of relevance, and we also have the network of laboratories. So in terms of the scientific information on the BCH, we have records specific to LMOs, genetic elements, organisms, biosafety virtual library resource records, laboratories for the detection and identification of LMOs, risk assessments generated both by independent processes as well as national regulatory processes, and our biosafety experts. So in terms of the amount of content on the BCH, we have over 17,000 records on the BCH, over 940 LMO records, over 840 genetic element records, over 260 organism records, over 70 laboratory records, over 1,500 virtual library resource records, over 2,600 risk assessments, and over 360 biosafety experts. So in terms of how the information actually looks like on the BCH, I will use a living modified organism record as an example. So generally the records are divided into a few parts. So the first part at the top here for LMOs includes our quick links. So this is a QR code that is scannable by a smartphone and allows you to access, it brings you directly to the BCH. It includes a label with information on the OECD unique identifier if it's available and the trade name. Following this, you have the sort of more general information on the LMO. So you have the trade name if it's available, the transformation event, the OECD unique identifier, followed by information on the developer, including contact information if available, a general description of the LMO record. Generally, this includes information sort of on what the LMO has in terms of traits. We have a link to a specific organism record, so in this case it's a cotton, and then we have a point of collection field. Generally, at the Secretariat, this could be a geographical location, if it's applicable, or if there's information on a particular cultivar or variety of plant or animal, we'll include it there. Following this, we have more of the molecular details. So we have information on the vector, the modification process used. Then we have a genetic elements construct. So us at the Secretariat, we reconstruct how the genetic elements would essentially look like in the LMO itself and include base pair sizes if it's available. And then we have all the genetic elements that are linked to this record. Following this, we have a field where we make sure that we include information on how the genetic elements interact within the LMO and how they interact with each other, information perhaps on the vector, or if the developers provide a sequencing analysis or bioinformatic analysis. We include some of this information here to sort of help understand the molecular biology of the organism itself. Following this, we have the modified traits. So if it's herbicide resistance, or herbicide tolerance, or resistance to an insect, the common uses for the LMO. And then we have our detection methods field. So here we have automated links to the European Union Reference Laboratory for GM food and feed, and the CropLife International Detection Methods database. So these are automated links that are automatically displayed on the LMO records, and we'll take you to those websites so you can get information on detection. Following this, we have an additional information section, as well as we include other relevant information, whether it's the Eugenius website, or if there's been regulatory documents released, or scientific publications. So we try to do our best to include as much information that we find in the LMO records. And then at the bottom of the records, and all records on the BCH, we sort of have this cross-referencing section. So here, all the records that reference this particular record are displayed here. So in this case, we have other living modified organisms, such as involved in stacked events. We have decisions, risk assessments, as well as laboratories. So if you are interested in, say, finding out which laboratories can detect and identify this LMO, you can expand, and then you can sort the information based on the last updated or the particular name. So in terms of the other records and the global overlook of the BCH and detection-related information, for LMO records, we have our, as I've shown, our detection methods field. We have the molecular biology part of it, the characteristics of the modification process. And then at the end, in the additional information section, we include as many publication sequences as whatever publicly available information that we find available will be included here. For genetic elements, we always include the name that we work with the Eugenius database on the GMO genetic elements thesaurus to have a standardized name for all genetic elements to facilitate interoperability between databases. And I also try to include sequence information if it's available on GenBank or UniProt. For organisms, it's similar. If the genome is available, we'll include it on the record. For biosafety virtual library resource records, these are scientific records. So we will include them on the BCH, and there's a particular section, a thematic area for detection, which will facilitate searching, which I'll show in a little bit. For the laboratory records, there's a wealth of information on the services that are provided, the methods used within the laboratory, the types of LMOs analyzed, the specific LMOs, the specific genetic elements, and then information on accreditation. And then we also have risk assessment records. So on these records, there's a field for proposed methods for detection and identification of particular LMOs. And then we also have our biosafety experts, and sampling and detection is one of the expertise fields. So to find information on the BCH, you go to our new website, bch.cbd.int, and then you can click on Search and select Records. And then under Record Types, which the default is to bring you here, you can then select the particular record that you're interested in. So for example, if we're interested in a laboratory for detection and identification, and we select it, it'll automatically filter out all the other records in the BCH and only show you the laboratory records. And then from here, if you're looking to refine your search, you can use the general search bar just as you would with Google. But we also have our sub-filters, which are specific to the common format. So these are predefined searches that based on the particular boxes that are checked and the information that's available. So again, we have a free text box here, which sort of works like Google. It'll search all of the text within the record for whatever keyword that you put in. And then we have for other types of filters, such as the services and activities, the types of LMOs, a particular geographical region that the lab is attributed to, the types of methods that are used, the particular LMOs or genetic elements. And this is actually an improved search function compared to the previous BCH. So you're able to, for example, exclude particular searches, or there's a lot of, if you're really savvy in this area, you can really refine your search. So for example, if you were to see what this looks like, if we were to look at the particular methods that are available, it comes with a predefined list, which you're able to select. So for example, if I was interested in a qualitative PCR, it'll automatically search the laboratory records, and all the records that reference that particular method will be displayed here. And then you're able to actually sort the search, whether it's last updated. You can also change the view, and it'll group the records in a particular way. You're also able to share this search with a colleague or another expert, and you can also export this search result as an Excel file. But I would also like to highlight that there's this Save This Search function, which allows you to actually, in your BCH account, save this search. So you can actually it'll be displayed, you can automatically go back to it and see if there's been any updates. But if you're worried about missing updates, you can also select the Send Me an Email. So anytime a record is added to the BCH that matches your search criteria, you'll be emailed an alert to check the BCH. So in terms of finding other ways of finding information on the BCH, there's this cross-referencing between records. So if you're interested in a particular LMO, and then you wanted to see information on a particular genetic element, you could go and find the genetic element and click on it, and it'll lead you to the particular record. Or if you're interested in a laboratory for detecting and identifying, at the bottom of the record, you'll see all of the records that are cross-referencing. So this cross-referencing within the BCH is really important, because perhaps you have an idea of what you're looking for, but don't really know how to search for it, so you can go through the records themselves and also find the information that you're looking for. Another way to find information is through our registries. So we have 3 registries on the BCH, living modified organisms, organism registry, genetic element registries. So this is a list of all of those specific type of records on the BCH for you to scroll through and select as you need. So moving forward, following the launch of the new platform in November 2021, the Secretariat is working really hard to improve the BCH and to ensure and maintain its operation. In the recent decision on detection and identification, CP 1011, parties were invited to submit information on their laboratories using the laboratory common format, and we will be following up with parties to improve the list of laboratories on the BCH. We're also exploring other opportunities to collaborate with other databases and improve the interlinkages between the BCH and other databases. So currently we have our JRC GMO methods, crop life detection methods database, FAO GM food platform, OECD BioTrack product databases, but we're looking for more ways that we can improve the BCH and provide really as the BCH is a one-stop source for all of the compiled information on biosafety. So to engage with the BCH, I encourage you all to sign up for a BCH account. It's free. When you go to sign in, you just click create an account, and it's an automated process. You put in your information and you can register for the BCH. I also encourage you to submit information to the BCH. So you're all experts, you're working in labs, you have high impact publications. We would love to receive them. We encourage you to submit them to the Secretariat so we can make it available on the BCH. We also encourage you to provide information on your laboratory, to improve our list of labs on the BCH. But we're also looking for feedback. So if you're using the BCH and you have a particular point that you'd like to raise with us, we encourage you to reach out to us. You can also get help when using the BCH. We have a new chat function at the bottom of the page, but you can also click on the question mark in the top right-hand side of the page, and you can access our knowledge database and access training materials on how to use the BCH. So with that, I would like to thank you for your attention yet again, and you can reach the Secretariat at any of the emails above or websites or social media. Thank you.